unwind and separate. Okay, so the separation of the two DNA strand is catalyzed by this enzyme, which is helicase. So the function of helicase is to separate these two double-stranded DNA so that it separates and become template. Okay. So this is the region where we call it as replication fork. So we have one here, one replication fork, and the other one here. Okay, so this is the formation of the replication bubble. So replication fork will be at both ends, okay, of the replication bubble. Okay, so it is a Y-shaped region where the parental strands of DNA begins to unwound. Some of the protein involved during the initiation of DNA replication are these. So I've mentioned to you why is it called protein, because most of it are enzymes, but we have here where we call it a single strand binding protein. It is not an enzyme. It is a group of protein that the function of this protein is to make sure the two double-stranded DNA maintains separate. It doesn't rejoin. Okay. So the synthesis of the synthesis of new DNA strands will start from the 3' end of the RNA primer. Okay, so as you can see, to initiate the process of DNA replication, to synthesize the new DNA strands, the process requires the synthesis of RNA primer. So the synthesis of RNA primer is synthesized by this enzyme, which is primase. So why is RNA primer needs to synthesize first? Because in order to add DNA nucleotide, which is the enzyme that adds the DNA nucleotide, we call it as DNA polymerase 3. That enzyme, DNA polymerase 3, will only add DNA nucleotide at the 3' end of a primer, or the 3' end of the elongating DNA strand. Okay. So I've drawn to you this diagram last week. So let's go to this table. Okay. So protein involved in DNA replication. So let's look at the protein one by one. The first enzyme here is the helicase. Okay, the helicase. So these are the functions. So the first function is to first, okay, kita rearrange lah kan. So the first function is to unwind first. So the enzyme will unwind the double helix at the replication fork. Okay, so this is the replication fork. The helicase sits here. Okay, so it is to unwind the DNA double strand and then separate. So once the DNA unwinds, it becomes straightened. So then only the two DNA strand can be separated. Okay. So separates the two parental strand to become template. Okay, so what is template? It is the strand that is used to synthesize the new complementary DNA strand. Okay, so how to separate the two DNA strand? As we know, here you see that there's pairing between the bases, can between the bases. Let's say, for example, this is between adenine and thymine. So there will be formation. There will be hydrogen bonds between the two bases. So the enzyme will break the hydrogen bonds between the bases. Okay, so by disrupting the hydrogen bonds that holds the two strands together. So all the way here, the bonds that hold the two DNA strands are hydrogen bonds between the complementary bases. So the helicase will break, okay, the hydrogen bonds. You can use both words. You can use disrupt the hydrogen bonds, or you can use by breaking the hydrogen bonds, whichever terms you can use as long as the explanation is correct. Okay, so next, the second protein here is the single strand binding protein, SSB. So binds to the single strands of DNA just after the parental strand separation. Okay, so if we look at this diagram here, so this is where the SSB will bind. Yeah, so after the two, then it has been separated, so it binds here. So it is to make sure that the two strands remain separated. Okay, so and then stabilizes the single strand DNA and prevents the formation of the helix before it can be used as template for replication. And then we have next the enzyme, which is topoisomerase. Let's look at the diagram here. So this is the location of the topoisomerase, okay, ahead of the replication fork. So The function of the enzyme is to prevent excessive coiling, relieves overwinding strain ahead of the replication fork by breaking, swiveling, and rejoining DNA strands. So this one I've explained to you already on the analogy of a spring. Okay, so imagine you have a spring and then you try to straighten the spring, so you will observe that there is a great pressure, okay, ahead of where the region that you straighten, okay. So that pressure, you need to reduce the pressure, okay. So that is the function of the topoisomerase, relieves overwinding strain ahead of the replication fork, okay. So and then so you have to really know the explanation lah, kan. Kadang-kadang dalam answer scheme tu it requires you to 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 write the full answer. If you just, for example, the question asks you, what is the function of topoisomerase? Relieve overwinding strain. That is your answer. So you may not get the full mark, or you may not get the mark. Okay. So you have to explain. Okay, the function of the topoisomerase is to relieve overwinding strain ahead of the replication fork by how? By breaking, swiveling, and rejoining DNA strands. Okay. So and then we have the next enzyme is DNA primase. You look at the name of the enzyme, it is DNA primase, but it is to synthesize RNA primer. So do not write the enzyme as RNA primase, it will be wrong. Okay, so the enzyme is DNA primase. It is to synthesize RNA primer at the 3' end of leading strands. Okay, so I will explain to you later the features of leading strand. We have two types of strands in the synthesis of the new DNA strands. Can. So we have here the leading strand, the other one is the lagging strand. Okay. So so it is to go back. So it is to synthesize RNA primer at the 3' end of the leading strand at the and at the 3' end of the Okazaki fragment of the lagging strands. Okay. So if you notice here, the synthesis of RNA primer is it can start, or it will start at the 3' end. Sorry, at the 5' ends. Okay. So it starts with a 5' end first. Okay. So that's why for the DNA synthesis, RNA primer is needed, okay, to be synthesized, because it can be synthesized starting with the 5' end, okay, and producing a 3' end, right? And then, so next one, so the DNA primase uses the parental DNA as template, okay, to synthesize a short RNA primer, all right, and that consists of 5 to 10 nucleotides long. Okay, so the primer is just short. It consists of only 5 to 10 nucleotides. The nucleotides added here is RNA nucleotide. So you should be able to know by now the difference between the RNA nucleotide and also the DNA nucleotide. Okay, so one obvious difference between DNA RNA nucleotides is the type of 5-carbon sugar. For the RNA nucleotide, it is the ribose. Can. So for the DNA nucleotide, it is the deoxyribose, right? So and then also the basis for RNA, you have uracil, and then for DNA, you have the thymine. Okay. So it uses DNA primase. It uses the parental DNA to synthesize short RNA primer by adding RNA nucleotide one at a time. Okay. So and then we have here, okay, so this is the enzyme that is responsible to add the DNA nucleotides. Okay, DNA polymerase 3. You look at here, okay, beneath here we have DNA polymerase 1. So make sure you know the difference between DNA polymerase 3 and also 1. Okay, so for DNA polymerase 3, its function is to use the parental DNA as template to synthesize new DNA strand by covalently adding DNA nucleotides to the 3' end of pre-existing DNA strand or the RNA primer. So as you can see, in order to add DNA nucleotide, it is added by this enzyme, but this enzyme can only add DNA nucleotide where there's a 3' end. Okay, the 3' end can either be located on RNA primer, okay, or the elongating DNA strands, okay. And then for DNA polymerase 1, can so the function of DNA polymerase 1 is to remove the RNA nucleotide of primer from the 3' end and replaces with DNA nucleotides. Obviously, can because we are synthesizing DNA, so we do not want the RNA nucleotide in the RNA primer to remain, can, in the DNA molecule. So that RNA nucleotide have to be removed. Can. So the removal of that RNA nucleotide is done by the RNA polymerase 1 and replaced with DNA nucleotide. So and then, all right, and then we have the next enzyme, which is DNA ligase, right? And so the function of DNA ligase is to join, or anneals. Anneals is to join lah, kan, to make sure that Okazaki fragments will be attached to one another to form lagging strands. Okay, on the leading strands, join the function of DNA ligase is to join the 3' end that replaces RNA primer to the rest of the leading strands of DNA. Okay, so this one here, can, so then the this is this the term Okazaki fragments. It is if you look at here, can, so this here, as you can see, I've drawn this already, right? So this is replication bubble, right? So this is origin of replication. So we have both template will have origin of replication where the DNA synthesis, DNA will be synthesized. Okay, new complementary DNA will be synthesized. So as you can see here, from the origin of replication, on one of the each of the template strand, there will be the synthesis of the leading strand. Okay, both templates. So here is the synthesis of the leading strand for this template. Okay, but as you can see over here, there is the synthesis of DNA strand that are synthesized as fragments. Okay, so this fragment, we call it as Okazaki fragments. They are synthesized in fragments. Okay, and then as you can see from this diagram also, the red line here is indicating it is the RNA primer. Okay, so the RNA primer will be later removed and replaced, okay, with DNA nucleotide by DNA polymerase 1. So once you have removed all this primer and replaced with DNA nucleotide, you have to join all these fragments. Okay, so to join all these fragments, you have to join using DNA ligase. Okay, so the joining of Okazaki fragments here, each fragment we call it as Okazaki fragments. Okay, so the joining of Okazaki fragments by the DNA ligase, it forms lagging strands. Okay, lagging strands. And then once lagging strand has been synthesized, okay, a continuous lagging strand have been synthesized, you have to join the lagging strand to the leading strand. Okay, so this produce a continuous DNA, new DNA molecule that is complementary to the template, okay, DNA template here. Okay, so that is the function of DNA ligase. All right. Okay, so we're done with this table. Okay, so to synthesize a new DNA strand requires the enzyme DNA polymerases. Okay, so DNA polymerases cannot initiate the synthesis of a polynucleotide. All right, so the DNA polymerases catalyzes the synthesis of new DNA strand by adding nucleotide, okay, DNA nucleotides to the existing 3' end of an RNA primer, right here. And so primer, ya, gambar ni. Primer is synthesized. So this is the 5' end of the primer. So here is the 3' end of the primer. So once a 3' end has been synthesized, then only the DNA polymerase 3 can add DNA nucleotides. Okay. Okay. So RNA primer. So the DNA polymerase will add the DNA nucleotide to the existing 3' end of the RNA primer, or the growing DNA strand, and never to the 5' end. Okay, growing DNA strand is here, can, here, Okay, so this is the growing DNA strand. So as you can see, as the new DNA strand elongates, okay, here there is a 3' end. So this is where the DNA polymerase tree will add the next and the next and the next DNA nucleotide. Okay. Okay, so next we have here another component, or another component, like another um component lah, we call it as, okay, call it as a component. Okay, so molecule that is required for synthesizing new DNA strands is the building blocks, okay, the monomers, right? So the monomers is, we call it as nucleoside triphosphate, or DNTPs, right? So these are the building blocks for DNA replication. So there's four types of DNTPs, which we have: DATP, DGTP, DCTP, and DDTP. Uh, um, N is referring to the term nucleoside lah. Okay, so this is adenine, guanine, cytosine, thymine. Okay, for adenine, for guanine, cytosine, and thymines. These are the building blocks that is required to synthesize DNA molecule. Okay, so for example, right, later in chapter 2, we are going to look at a process, we call it as PCR, polymerase polymerase chain reaction. Okay, polymerase chain reaction. So for that process to occur, you need a sample of DNA. Okay, so a sample of DNA is placed into a test tube, into a small tiny tubes. Okay, a small tiny tube. So in order to the purpose of PCR is to replicate DNA in vitro in a machine. So ingat balik masa contohnya musim COVID dulu kan, in order to detect whether or not you have the virus, kan, so as a sample from your saliva, or from your nostrils, you get a sample of the mucus, and then that contains the DNA, kan, so your samples of DNA. So you want to know whether in your DNA there is the virus RNA, the virus genetic material, so that you place into the test tube, kan, so your sample, kan, that may contain the virus, the genetic material, and then you add all those enzymes that I've mentioned here. You add the DNA ligase, the polymerase tree, kan. So and also in order to synthesize new DNA molecule, you have to add the DNTPs, kan. So the machine will run and then replicate the DNA, the genetic material lah for the virus. Virus doesn't have DNA, okay? It has RNA. But anyhow, the machine will replicate the genetic material so that we can detect whether or not the sample contains the virus. Okay, so that is the machine, we call it as PCR lah. Okay, so PCR to replicate the DNA. So in order to replicate the DNA, high temperature will be applied. So as you can see, as we know, kan, DNA molecule, it is double stranded. And it is double stranded due to the hydrogen bonding between the bases adenine, thymine, guanine, cytosine, kan. So by increasing the temperature, you break the hydrogen bonds. Okay, so okay, so that is on the building block lah. Okay, so let's pause for a minute. If you have question, you can ask. I am going to switch on the aircon. Kalau dengan lecture you will observe that saya akan ambil masa sikit untuk pada macam contohnya 8.30 kan, kalau ada kelas 10.30, dia ada masa yang mana aircon dalam bilik ni akan ditutup dan saya kena on the mic. So kejap. Okay, so next, alright. Next slide. Um, Okay, so RNA replication, anti-parallel elongation. So the synthesis of new DNA molecule during DNA replication involves two types of strands. The first type of strand is the synthesis of the leading strands. Okay, so this is the leading strand synthesized on both of the template. So the feature of the leading strand is that the synthesis will start from the origin of replication towards replication fork. Both. From origin of replication, it is synthesized towards the replication fork. And then for the synthesis of the lagging strands, the feature of the synthesis of lagging strand is that it is synthesized from the replication fork towards origin of replication. Okay, from replication fork towards origin of replication, but it is synthesized in fragments. Okay, so both on both sides from replication fork towards origin of replication. Okay, so I will later we will see that why is this strand is synthesized in fragments. Okay. Okay, let's look at first for the synthesis of the leading strand. Okay, so the first point here again. So along one template strand starting from the origin of replication here. Okay, that has a 3' end. So the DNA primase or the primase synthesize a short RNA primer. If you are not sure or you are confused during the exam lah, the test kan, also whether it is DNA primase or RNA primase kan, so because it is synthesizing RNA primer, maybe you may make careless mistake by writing RNA primase kan. So in order to avoid that, you can also write the enzyme as primase only. Okay, so primase. So primase synthesizes a short RNA primer. So here is the origin of replication on the template strand. So here is where the primase synthesizes the RNA primer. So the synthesis of RNA primer will start with 5' adding the RNA nucleotide one at a time, producing a 3' end on the primer. Okay, so once a 3' end has been produced, so here it produces a 3' end on the primer. So then only the DNA polymerase 3 starts to synthesize a complementary strand continuously. Okay, so another feature of leading strand, it is synthesized continuously. It is not synthesized in fragments. It is synthesized continuously from the origin of replication towards the replication fork. Okay, so that's why it is highlighted in red. So this is the keyword that you have to take note. It is synthesized continuously by elongating the new DNA strand in the 5 to 3' direction. Okay, towards the replication fork. So these are the feature of the leading strand that you have to take note. It is synthesized continuously. It is elongated in the 5 to 3' direction. It is synthesized from the origin of replication towards replication fork. Okay. So another feature of leading strand is that it only requires one primer. Okay, one primer to synthesize the leading strand. All right. So and then next after the DNA the DNA polymerase 3 adds the new DNA strands by adding the DNA nucleotides, then we have to remove the RNA nucleotide in the primer. Okay, so to remove this RNA nucleotide is done by the DNA polymerase 1. Okay, so DNA polymerase 1 replaces the RNA primer with DNA nucleotide. So you do not want the RNA nucleotide, the enzyme will replace the RNA nucleotide with DNA nucleotides. Alright, okay, so after RNA primer is made here, the same explanation can. So after RNA primer is synthesized, you can use both words, okay? So the DNA polymerase 3 starts to synthesize the leading strand. Yeah, so next leading strand is elongated continuously in the 5 to 3 prime direction. 5 from the origin of replication to the 3 prime direction, okay, which is the here is near to the replication fork, okay. So this is the enzyme. You can so this is the enzyme lah. This is the enzyme which is DNA polymerase 3. You can add in your notes here to make it clear. This is the enzyme DNA polymerase 3. This is SSB. This is helicase, right? So over here you will have the topoisomerase, okay. Okay, so now we are going to look at the synthesis of lagging strands, okay, which are synthesized as fragments, where it is called as Okazaki fragment, okay. Okay, so as you can see, okay, we're done with the explanation on the leading strand. Okay, so we know that for the leading strand, it requires one primer. It is synthesized from origin of replication towards replication fork. It is synthesized continuously and from 5 to 3 prime direction on both strand here. Yeah, so here and here is the leading strand. But for the lagging strand, it is a bit complicated, okay. So because you have to imagine, the actual process of how the DNA replication occur, that involves the replication bubble getting bigger and bigger and bigger, okay. So okay, for the synthesis of lagging strand, it is synthesized discontinuously. This is one feature of lagging strand. It is synthesized discontinuously compared to the leading strand. It is synthesized continuously. For the lagging strand, it is synthesized discontinuously as short Okazaki fragments, okay, in the direction away from replication fork. Okay, so you can write again, or you can give the feature of the lagging strand. It is synthesized from or replication forks towards origin of replication. Okay. Okay, so this is the process, right? So the same as the synthesis of the leading strand, where the process is initiated through the synthesis of RNA primer first, which is done by the primase. Okay, so primase joins RNA nucleotide to a primer. So imagine, okay, so not imagine, so this is the origin of replication, okay? So this is origin of replication. Here you see is the primer of the leading strand here. This is the region here, dia besarkan. It is, this diagram is the enlarged diagram from here, okay? So here is the primer of the leading strand over here, okay. So we are starting over here at the origin of replication. So here we are going to observe the synthesis of the first Okazaki fragment, okay. So this is the first Okazaki fragment. This is the second Okazaki fragment that is synthesized. This is the third Okazaki fragment. So as the replication bubbles get bigger and bigger, you will observe that there's more synthesis of the next and next and next Okazaki fragments. The first Okazaki fragment is synthesized at the or near the origin of replication. Okay, near the origin of replication. Same goes to over on this side. So this is the first Okazaki fragment, the second Okazaki fragment, the third Okazaki fragment, and as the replication proceed and the replication bubble gets bigger and bigger, kan, dia makin besar, bergerak, anjak ke depan, kan. So you are going to observe, so this one here, as the replication bubble opens, kan, so it synthesizes the fourth. As the replication opens more, fifth, sixth, kan, and so on, right? So right, okay. Okay, so once the primer has been synthesized, okay, so this is, as you can see, you are going to observe that, again, the synthesis of the primer, it doesn't start exactly at the origin of replication. It waits for the replication bubble to get to a certain distance, okay, so then only the primer will be synthesized. The primer will not be synthesized at the origin, but it is synthesized like further away from the origin of replication, which is somewhere here. Okay, So and then okay, so once the RNA primase, RNA primer is synthesized, so a 3' end is available for the DNA polymerase 3 to add DNA nucleotide to the primer to form the Okazaki fragments. So as you can see, Okazaki fragments is synthesized towards origin of replication, which is the synthesis of the DNA will be from 5 to 3 lah, kan, for 5 to 3. Because the enzyme DNA polymerase is used, can DNA polymerase 3 is used, DNA polymerase 3 can only add at the 3' end. Over here, you observe that there's a 5' end of the primer of the leading strand over here, right? So once the Okazaki fragments has reached this region, the synthesis of the Okazaki fragment will be terminated. Sebab apa? Sebab dia jumpa the DNA polymerase 3, it encounters a 5' end of the primer over here. DNA polymerase 3 will not add at the 5' end, only at the 3' end. So it stops here. So as you can see, this produces the first Okazaki fragments. Okay. So DNA polymerase 3 detaches once it meets the RNA primer to the right here. So this is the RNA primer, for example, of the leading strand. Same when the second fragment is synthesized, kan, the primase synthesizes the primer, DNA polymerase 3 as DNA nucleotide. So once it meets the 5' end of the primer of the first Okazaki fragment, the DNA polymerase 3 will stop, okay, producing the second Okazaki fragment. Same goes to the synthesis of the third Okazaki fragment. The primase synthesizes the primer from 5 to 3' end. Once it meets the primer of the second Okazaki fragment, the process stops. Okay, so it is synthesized in fragments. Okay, so fragment 2 is primed. Okay, is primed, where the DNA polymerase 3 will add the DNA nucleotide, and then detach once it encounters the it reaches the fragment of the first primer. Okay, sini kan. So this is the synthesis of the second Okazaki fragment. Primer has been synthesized. DNA polymerase 3 adds nucleotide, add another nucleotide, add another nucleotide, add another nucleotide, kan, of the Okazaki fragment, and it meets, okay, the enzyme. It encounters the 5' end of the primer of the first Okazaki fragments. So once the DNA polymerase 3 encounters the 5' end of the primer, it then detached. Okay, it then detached from the Okazaki fragments. Okay. Okay, so next, this should be highlighted as red as well. Kan? So it is DNA polymerase 3. Kan? So you do not want RNA nucleotide in the DNA. Kan? So the function of DNA polymerase 1 is to degrade the RNA primer. Kan? You replace all these red nucleotides, all the RNA nucleotide with DNA nucleotides. Replace with DNA nucleotide by DNA polymerase 1. Okay. So you have the first fragment, the second fragments, the third and the fourth, kan? So all that fragments has to be joined together by DNA ligase. Okay, so the joining of Okazaki fragments by DNA ligase is to form the lagging strands. Okay, into continuous DNA strand. Right. Okay, let's look at this diagram. Let's not diagram, this animation. Okay. So next slide. Okay, so that animation, it shows you the synthesis of the Okazaki fragments and then joining the Okazaki fragments to form a continuous lagging strand. Okay, so let's look at this diagram. Okay, so as you can see, so this is the replication bubble. Okay, so you see here is the origin of replication. So you have to imagine as the DNA molecule is synthesized, you have to imagine that the replication bubble will initially start small. Okay, so it starts small. So the small, the region where the DNA replication is formed, where the replication bubble is start small, that is when the first Okazaki fragment synthesize. Kan dia akan buka dekat origin of replication, buka, and then it produces the first Okazaki fragments. Buka lagi, the replication bubble gets bigger, second Okazaki fragment synthesize. Open more, kan, the replication bubble gets bigger, the third Okazaki fragment synthesized and bigger. The fourth and the next Okazaki fragments are synthesized, will be synthesized. Okay. All right. So let's look at this Venn diagram here. Okay. So this is just to recap. This is the overview of how the DNA replication occurs for both leading and lagging strand. Okay. So initially it starts at the origin of replication, and then to unwind and separate the DNA strand, it is done by the enzyme helicase. Okay, helicase unwind the parental double helix. Okay, here it is located at the replication fork. So and then we have another type of protein, we call it as SSB, single strand binding protein, which function to stabilize the unwound parental DNA. Okay, so the parental DNA is separated to prevent the two parental DNA from rejoining, from forming helix again. So it is stabilized by SSB. The two strands are stabilized by the SSB. So and then we have here okay, the synthesis of the leading strand. Okay, so the synthesis of the leading strand, it starts at the origin of replication towards replication fork. It is synthesized continuously in the 5 to 3 prime direction. 5 to 3 prime direction. Okay, by the enzyme DNA polymerase 3. So and then we have here the synthesis of the lagging strands, which are synthesized as Okazaki fragments. As you can see here, it is numbered, that the Okazaki fragments are numbered. The first fragment, the second Okazaki fragment, the fourth Okazaki fragment, the third, the fourth, kan. The fifth that is starting to be synthesized, kan. So the lagging strand are synthesized as fragments discontinuously in also in the 5 to 3 prime direction. Any synthesis of nucleotide, polynucleotide, whether it is lagging strand, whether it is leading strand, whether it is RNA molecule, then the synthesis is always from the 5 to 3 prime direction. Okay, so and then we have here, we observe here, there's the primase. Okay, so the primase will synthesize the RNA primer. Okay, so primase synthesize RNA primer, which is extended by the DNA polymerase 3 to form an Okazaki fragments. Okay. So then all the RNA primer will be removed and replaced with DNA nucleotide using DNA polymerase 1. This should be bold, DNA polymerase 1. So next, okay. So and then all the Okazaki fragments will be joined by the enzyme DNA ligase. Let's watch this animation first. Yes, question. Buku Fatin, Hai Sofia. Do you have question? Mana tadi angkat tangan?
No, Madam Administrator silap tekan.
Okay. Anyone else? Do you have any questions? Before we proceed with the next subtopic, so subtopic, the next one is on photosynthesis. So in order for us to proceed with the photosynthesis, you need to understand DNA replication first. Any question for this one? For DNA replication? Tak ada soalan, saya tanya nanti. Laila Najiha. Kat situ. Mana?
Yes, madam.
Okay. Saya nak tanya, okay, for the enzyme DNA ligase, it is the enzyme that catalyzes the joining between Okazaki fragments or the joining between the leading to the lagging strands. Okay. So name the type of covalent bond that is formed that joins the fragments together. The name of the bond. It is a type of covalent bond. What is the name of the covalent bond that attaches the nucleotide together in the Okazaki fragments or from leading to lagging strands? Apa nama covalent bond tu? What is the name of the covalent bond that is catalyzed by DNA ligase? Tengok gambar ni kan? Right? Kan? So imagine the red nucleotide here, the RNA primer, has been removed and replaced with the blue one, which is DNA, kan? So then you observe the DNA ligase will come and join, kan? The Okazaki fragments, and then from the you join the Okazaki fragment to form the leading strand. You are going to join the leading to the lagging strand, kan? By the DNA ligase. What is the name of the bond that is formed used to join all the DNA strands together? What is the name of the bond?
Is it a phosphodiester bond?
Betul lah. Kan? So the name of the bond is phosphodiester bond because phosphodiester bond is the bond that joins the nucleotide together. Dia daripada kefahaman mana? Kefahaman yang kat sini. Kan nampak? The phosphodiester bond, they are the bond that links the nucleotides together. That is the function of the DNA ligase to catalyze the synthesis of the phosphodiester bond to join the Okazaki fragments together or to join the leading to the lagging. Okay, so good. Saya nak tanya lagi soalan. Since you don't have question, then I will ask. Wanu Shaza. Okay, saya nak tanya kan. Kenapa Okazaki fragments ni dia disynthesizkan as fragments? Kenapa dia disynthesizkan daripada replication forks towards origin of replication, dan kenapa kalau yang dekat sini is the first, bukan sini yang satu, yang kedua, ketiga, keempat, kenapa yang first dekat dengan origin of replication? This is the first, second, third. Kenapa bukan 1, 2, 3? Kenapa 1, 2, 3? Why? Dalam kereta ke apa? Dalam kereta ke?
Tak, saya tengah fahamkan lagi.
Okay. Faham tak soalan saya? Kenapa? Why is it that the Okazaki fragments it starts here, the first fragment, the second, the third. Kenapa bukan this is the first, this is the second, this is the third? Anyone? Do you have any clue why? Would anyone try to explain kenapa? Any volunteer? Kalau boleh explain tu, then you know that why is the lagging strand synthesized in fragments. Kenapa agaknya the lagging strand are synthesized as fragments? Kenapa? So ini saya nak terangkan kenapa Okazaki fragments tu kan, ataupun the lagging strands are synthesized as fragments. Okay. Purple ni saya draw Purple saya draw untuk indicate that it is the template strand. Okay, so this is the replication bubble. Kita start balik. Okay, kita imagine So this is the DNA molecule. Okay. So this is the origin of replication. Okay. So now in order to synthesize or in order for DNA replication to occur, you need to separate the two DNA strands. Okay. So you have to separate. So you separate and then it will start to form a replication bubble. Okay, let's number the strand. So 5, 3, over here is 3, over here is 5. Okay, same goes to this one. This is 5, this is 3, this is 3, this is 5. Here you have the origin of replication. Okay, so at the origin of replication, awak kena tahu the numbering also kan. Bila sebelah sini kita ada 5 kan. Kita imagine bila kita potong kat bahagian sini, over here we have a 3' end. Okay, a 3' end. So bila kat situ sini 3, awak kena imagine juga sini pun 3 juga. Macam tu. Kita kena imagine kita potong dia. Macam tu lah. Potong dia tu siapa? The template lah. Okay. So now, okay, in the synthesis of the leading strand, there's no problem to understand the process. Okay. So kita nak synthesize yang dia punya primer dulu. So primer start dengan 3 and then 5. Sorry, start dengan setelah kan the primer because atas 3 bawah 5. Hilang dia. Okay. Sini kita akan menghasilkan the 3' end. So same goes to here. Kita akan synthesize the primer 5, 3. Okay, leading strand dia tak ada masalah sebab once we have synthesized the primer, it produces a 3' end, then DNA polymerase 3 can come and add DNA nucleotide, dia tambah. DNA polymerase 3 tambahlah nucleotide. Kan? So the elongation of the leading strand is continuous from origin of replication towards replication fork. Okay, replication fork. From origin of replication towards the replication fork. Leading strand has no problem. But for the lagging strand, it is synthesized as fragments. Okay, so imagine, so we have initially the replication bubble is small, it starts small. Okay, so imagine here kalau Okazaki fragments, it is synthesized the same in the 5 to 3' direction. So sekarang ni kita nak cari pada template yang dilabel sebagai 3. Okay, yang dilabel sebagai 3 supaya kita boleh hasilkan RNA primer dulu dengan RNA primer kita nak hasilkan dia sama juga in the 5 Kan? 5 to 3 prime direction. So sekarang ni, dekat mana yang kita boleh hasilkan RNA primer ni? Kita nak tengok dua region yang kosong ni. Sekejap Kan? Ni, kita nak synthesize the Okazaki fragment. Tapi mana nak letak RNA primer ni? Kita nak cari kat mana kita boleh synthesize the 5' end. Okay, so therefore, kita nak hasilkan RNA primer tu dekat bahagian, nampak kat sini. Bila kat sini 3 kan? Dekat sini pun pada template tu is also 3. It is also 3. Pada template tu is also 3 prime. It is also kat sini kan pada template it is also 3. Okay. So bila 3 kita bolehlah complementkan dengan 5. Right. Kan? So kita complementkan dengan 5. So this is the synthesis of the primer for the first Okazaki fragment. So atas ni 5, hujung ni 3. Okay. Sini 3 pair dengan 5, 5 hujung sini ada 3. So you have produced a 3' end on the RNA primer, then only the next enzyme can come, which is DNA polymerase 3 can come and add DNA nucleotide. So DNA nucleotide is added at the 3' end of the primer. So DNA polymerase add sama juga. Towards kan, the synthesis of the Okazaki fragment is from the replication forks towards the origin of replication. Ini nanti awak tanya soalan sekejap So this is the synthesis of the first Okazaki fragments. This is the initial phase where the replication bubble starts small. So tadi apa? Replication bubble as the DNA replication proceeds, the replication bubble will get bigger and bigger kan. So replication bubble akan membesar. Okay. Membesar macam tu kan. So saya padam ni sebab dia take space. Origin, here is the origin of replication. Okay. So sekarang ni kita nak imagine the replication bubble getting bigger. Okay. Sama je tempat duduk dia kan. So this is the origin of replication. This is the origin of replication. So saya kena padam sebab nanti tak boleh muat. Okay. Yang biru tu is the origin of replication. Dah terpadam. Okay. Sekarang ni kita nak copy balik yang kita dah draw tadi kan. So kat sini saya copy balik. Okay, the primer of the leading strand, because the bubble is getting bigger, so the synthesis of the leading strand will proceed without any problem. Kan dia akan proceed je. Kan, so as the replication bubble get bigger, dia akan synthesize continuously, no problem. Kita guna light dulu kan. Okay, so the synthesis of the leading strand has no problem because as the bubble gets bigger and bigger, it can proceed from 5 to 3 prime. From 5 to 3 prime. Okay, but for the lagging strand, for the synthesis of the Okazaki fragment, kita copy balik yang first fragment tu tadi. Imagine the first fragment is here, that has been synthesized. Tadi tu kan? Dia stop kat sini. Dia stop kat mana yang dijumpa primer. Okay, stop. So this is the first Okazaki fragment. The first Okazaki fragment. The numbering is 5 to 3. Sama je. 5, 3. Okay, so now you have this region here that is empty. Due to the replication bubble that gets bigger kat sini. So this is empty. Sebab apa? Replication bubble tu dah membesar kan. So this one is empty, this one is empty. So now you are going to synthesize the second Okazaki fragment. So Okazaki fragment, the second one, so you look at the numbering of the template. 5 tadi, 3 kan? Ini 5, ini 3. So dekat sini, bila sini 5, bawah ni, sekejap. Bila sini 5 kan? Sini pun 5 juga. Bila sini, sekejap. Kan bukan situ 5, terbalik. Belah sini kan, situ 5, bawah, sini 3. 3 kan. So ini pun 3 juga. 3 pair dengan apa tadi? 3 pair dengan 5 kan. So the synthesis for the RNA primer for the second Okazaki fragment can be synthesized. This is the, saya repeat, this is the synthesis of the RNA primer for the second strand. Second strand, synthesize its primer. Then you produce a 3' end. Then only DNA polymerase 3 can come and add DNA nucleotide, synthesize from where to where? From replication fork towards the origin of replication. Kan? So lepas tu jumpa dengan 5' end of the primer, then the process stops. Same goes to here. Yeah, so RNA primer is synthesized and then it produces a 3' end, then only synthesize the RNA primer. Numbering is 5, 3, and then DNA polymerase 3 can come and add DNA nucleotide, tambah sampai dijumpa RNA primer of the first Okazaki fragment, then the process stops. This is the second Okazaki fragment. So you have to imagine the replication bubble will start bigger and there will be gap tadi, kan? So synthesize the third, the fourth, the fifth. So that's why the synthesis of the Okazaki fragment, the fragment that is nearer to the origin of replication is the first fragment. The Okazaki fragments yang paling dekat dengan replication fork is the latest one that is being synthesized at that point. Okay? Any question? Ada soalan tak?
Madam, saya nak tanya, apa hubungan Okazaki fragment tu dengan RNA primer? Saya macam confused sikit.
Apa hubungan?
Saya macam confused sikit.
Okazaki fragment adalah DNA strand kan? New DNA strand that is synthesized according to the template, using the template to synthesize DNA strand. Okazaki fragment tu sebab dia adalah DNA strand yang akan dihasilkan, yang akan menjadi new DNA molecule according to the template. So that's why in order to produce any DNA strand in the replication bubble, you have to synthesize the primer first. Dia tak boleh menonong-nonong start dengan start dengan DNA polymerase 3. DNA polymerase 3, faham tak? Tak boleh. Apa kaitan? Faham tak? Itulah kaitan dia. In order for Okazaki fragment to be synthesized, RNA primer has to be synthesized first. Saya draw lah bulat. Garis mula. This is the replication bubble. Okay. Origin of replication. Okay. So kita nak sekarang ni kan, you have to know the numbering on the template. Okay, you have to know the numbering of the template. So kalau untuk kita sekarang ni kita nak explain, kita nak tengok leading strand tak ada masalah, kita nak tengok the lagging strand. The lagging strand, it is synthesized as Okazaki fragments. The synthesis of Okazaki fragment, it is actually a DNA strand kan? The synthesis of DNA strand, it has to start with 5 to 3. Okay, sekarang ni kita nak untuk menghasilkan DNA molecule, kita nak gunakan DNA nucleotide menggunakan enzim apa? DNA polymerase 3 kan? DNA polymerase 3. So sekarang ni DNA polymerase 3 ni dia tak boleh nak initiate the process first. it must start with the synthesis of RNA primer first. Okay, so in order to synthesize the RNA primer, you have to identify where to synthesize the RNA primer of the Okazaki fragment that is going to be the lagging strand later. Okay, so here kan, apa feature Okazaki fragment? It is synthesized from replication fork towards origin of replication. Replication fork kat mana? Kita dah tahu dah kan, replication fork dekat area. Sekejap, mana ni? Dekat area sini. Replication fork. Okay. So Okazaki fragments, you have to synthesize the primer first. The primer is the same, it is synthesized in the 5 to 3 prime direction. Sekarang ni awak nak kena locate kat mana nak synthesize the 5 prime end. It is synthesized here. Here. 5, 3. 5, 3. Sebab apa? Sebab kat sini kita dah nampak kat sini pada template. Kita nampak pada template 3. So kat sini pun akan jadi 3. Pada template akan jadi 3 juga. Okay. Pada template kat sini. Ini terbalik ni sekejap . Balik je asyik-asyik ni. Sini 3 kan. 5, 3. Okay. So, bila kita nampak dekat hujung sini ada 3 kan. Sini pun 3 pada template tu. Sini pun 3 juga. Bila 3 baru boleh complement dengan 5 kan. Baru boleh complement dengan 5. So once we have identified where to locate the 5 prime end to synthesize the primer for the Okazaki fragment, so we are going to use the primase. Okay, so the primase will add RNA nucleotide to synthesize the primer of the first Okazaki fragments. Okay, so once a 3' end has been synthesized, then only DNA polymerase 3 can come and add the nucleotide to synthesize the rest of the Okazaki fragments towards origin of replication. Ini you have to imagine this is when, for example, the replication bubble starts small, kecil. So bila dia akan membesar kan, dia akan besar. So dia akan besar, let's say for example, replication bubble is over here, dia open up. Dia akan open up over here, macam itulah kan. So now you are going to synthesize the primase for the second Okazaki fragment. You are going to synthesize the primer for the Okazaki fragment yang kedua. Synthesize lah, kan. From replication forks was the origin of replication. Itulah kaitannya antara kenapa Okazaki fragment tu kena ada primer. Sebab Okazaki fragment tu is the DNA strand that is synthesized according to the template. Boleh? Faham? Tadi siapa? Sarah?
Boleh, madam. Thank you.
Okay. Ada soalan?
Madam, may I ask
Yes.
a question?
Ha.
Regarding sebab tadi you showed us a video where the DNA replication occurs until it reached to a point where the bacteria replicate sampai ada 2. So my question is,
Sekejap.
the
Mana ada bakteria ni?
the video that you showed just now.
Okay. Can you repeat the question please?
Alright, my question is to a certain point can the replication occurs to a point where it reaches where the bacteria replicates into two. So my question is, what is the initiation, as in the point where it separates into two, creating another bacteria tu?
Video yang mana refer pada bakteria? Sekarang ni kita tengok pada DNA replication. DNA replication tak kisahlah dia dalam bakteria ataupun dalam eukaryot. Ni. Nak refer pada mana sini Ke mana? Video yang mana
The video where it showed inside of the guy's stomach tu.
Oh, yang guys sambut tu tadi. Yang ni.
In both before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. The origin of result is here the DNA.
Soalan awak tadi up to what point? Up to what point tadi apa?
Up to what point does the replication occurs until it creates another bacteria?
Oh, yang itu takkan awak kena fahamkan. Kalau macam contohnya, kalau dalam bakteria ni dia berbeza dengan DNA contohnya manusia kan. Bakteria are prokaryotic cell. Okay, dia dah bagi contoh dalam okay, mana ni? Benda ni terus. Okay, so this is prokaryotic cell. Okay, beza dengan eukaryotic cell, macam contoh manusia kan. Our cell is much more complex. For prokaryote, E. coli for example, their DNA is smaller in size. Okay, for example, they have circular DNA, small size. But our cell is bigger, kan? Bigger and much more complex. Our DNA is much more longer. Okay, dia dua macam ni lah. Okay, so untuk prokaryote ni kita enlargekan dia punya DNA, circular DNA, or we can call it as plasmid. Okay. Circular ataupun. For this bacterial DNA or circular DNA, because the size is smaller, they only have one origin of replication. Okay, one origin of replication. Kat sini lah point dia, sini. Bukan kat bawah ni ini salah. The DNA only have one origin of replication. So for this kan, for the plasmid to replicate, to form two new plasmid, kan, inside a bacterial cell. So initially this is the parent, this is the daughter lah yang produce nanti kan. So in order for this to occur through the process of mitosis, okay, so the plasmid will open the DNA. Dia akan form satu kan. Starts to open the plasmid through the formation of replication bubble. Faham tak? Kat situ. Origin of replication. So kat sini yang mana yang kita tadi dah tengok tadi proses yang sama. Dan yang mana dia akan synthesize the leading strand kan, and also synthesize the Okazaki fragments to form the lagging strand. And so the Okazaki fragments synthesized in fragments. Okay. So as replication occurs, the bubble will get bigger, bigger, bigger in both directions. In both direction until apa until the DNA finish replicating to produce two daughter DNA strand. Dia akan proceed besar, dia akan jadi besar, besar, besar. Kena tengok balik, imagine. Tengok balik animation yang untuk eukaryotic cell ni. Yang mana? Kita ingat ada video dia dekat. DNA replication untuk prokaryote itu. Ni lah, dia tak ada animation dekat sini. Nampak ni? Okay. So the replication bubble will get bigger, 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 bigger until both the new daughter DNA molecule has been produced. Boleh faham ke? Tapi kalau untuk eukaryote, kan, because the DNA size is much more bigger, and so there will be more origin of replication. So as the bubble gets bigger, 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 at one point they will meet up, kan? And then when the replication bubble opens and they meet, kan? So it produce two new daughter DNA molecule, kan? So this produce complete and replicated DNA molecule in the daughter cells. Boleh?
Faham, thank you madam.
Okay, so can we proceed? So we are going to proceed with the next subtitle, because subtopic, not subtitle, subtopic. Okay, so I hope that you can look back, again, in your own time, the synthesis of the leading and lagging strand, especially on the concept of the synthesis of the lagging strand. So why is the strand synthesized as fragments? Okay. Okay, so let's look at the next subtopic, which is protein synthesis that involve two processes, which is transcription and also translation. Okay. Okay, so we in this subtopic we are going to look at the different type of RNA, and then basic principles of transcription and translation, genetic code, transcription of RNA, modification of pre-mRNA in eukaryotes, and translation, which is the process of synthesizing the protein. Okay, so the basic concept you have to know. I'm going to draw this in another slide. Okay. Okay, so imagine this is our cell. I am going to explain to you the basic concept of transcription and translation. Okay, how they are related to one another. Okay, how these two processes related to the function of DNA, later the DNA that leads to the synthesis of protein in your body. Okay. So your body is made up of many cells, okay? So in order for the cell to work, it depends mostly on the function of protein. So this protein can have many different functions. So like what we have looked at during last semester in the chapter of macromolecule, the subtopic protein. Protein can be enzyme, protein can be hormones, protein can be the transport protein that transport substances across plasma membrane. It can become antibodies, right? So protein in your body, in your cells, have many different functions. So that's why that is the role of the DNA in your cell. The role of the DNA in your cell is to store informations to make many proteins in your body. Okay, so basically the DNA in your nucleus is to store your genetic information. Okay, so genetic information. So for example here, we have a cell. We buat besar sikit. We have a cell. Okay. Inside this cell, we have the nucleus. Okay. So inside this cell, we have a nucleus. Okay. So the nucleus is where your DNA is stored. Nak saya nak buat dia lebih panjang. Ambil je. Okay. Okay, so this is just a fragment of the DNA inside your nucleus. Okay, a fragment. So inside your DNA, the function of the DNA is store your genetic information. Store your genetic information in the form of genes. Okay, so in your DNA there are many genes. So for example, this is the region for the first gene, second gene, third gene, fourth gene. For example, there are so many different genes. Okay. Gene 1, gene 2, gene 3, gene 4. Okay. So let's say, for example, the third gene here, okay, so the third gene, it stores the information that synthesize. For example, this is the cell this is the cell within a flower. Okay. So kita nak tengok sel dalam bunga. Okay, dalam pokok bunga. So this flower, it has, for example, what color? Let's say, for example, the flower is red. And then another phenotype of the flower, it can have a white flower. Okay. So the third gene here, it stores the information to synthesize enzyme, okay, enzyme that will involve in the process that lead to the synthesis of red pigment in the flower petal. Okay, enzyme that involve in the synthesis of red pigment that produces red flower. Okay, red flower. So so imagine this is the cell in the flower petal. Okay, in the flower petal, kelopak bunga. Okay, so only gene 3 will be activated and only the information within gene 3 is required, okay, to produce this enzyme. We call it as enzyme R lah, kan? Enzyme R, for example, that is synthesized, that is involved in the synthesis of red pigment later. So we only want the information from gene 3, okay? So because your DNA is really large molecule, it stores many other information, kan? So that is not required at that point, okay? So only information from gene 3 is required. Okay, so the DNA, it stores the information, but the process of synthesizing the protein, it occurs, we know that the synthesis of protein occurs in the cytoplasm, kan? Apa organelle? What is the name of the organelle that involved in protein synthesis? Is ribosome. Is ribosome. Okay, so ribosome is located in the cytoplasm. Kan? So ribosome is located in the cytoplasm. The synthesis of the protein should occur inside the cytoplasm, as there will be abundance of the monomers for the protein. Kan? We have abundance of amino acid in the cytoplasm. So how is that information from the DNA leads to the synthesis of protein in the cytoplasm that involves the ribosome in the cytoplasm and the amino acid also in the cytoplasm. So this is where the process takes place. First, transcription occurs to copy the information from the DNA into a smaller version. Okay, into a smaller version, we call it as mRNA, messenger RNA. Okay, so that information is copied into a smaller version. Then the mRNA will be exported out of the nucleus into the cytoplasm. So let's look at the first process of transcription. So transcription is where the information will be copied. The process is transcription. Copy the information. Okay, copy the information from the DNA into a smaller molecule which is mRNA. Okay, it copies the information to synthesize enzyme R. Okay, so mRNA is single strand, DNA is double strand. So kita dah belajar sem lepas chapter sel yang mana kita tengok apa the organelle nucleus. Nucleus they have nuclear pore kan, nuclear pore. So the nuclear pore is present kan, nuclear pore, kan. So the nuclear pore it allows the mRNA to exit the nucleus and find a ribosome inside the cytoplasm. So the ribosome, it has two subunit, the large subunit and the small subunit. The mRNA will go in between the subunit of the ribosome. The ribosome will read the information on the mRNA, and reading that information leads to the synthesis of protein. Okay, it leads to the synthesis of protein. So protein will be synthesized. In order to synthesize the protein, it requires another type of RNA, which is tRNA. RNA. What is tRNA? Transfer RNA. Transfer RNA will bring specific amino acid, okay, specific amino acid to the ribosome, that is read by the ribosome, kan? So the tRNA will bring specific amino acid according to the code, on the mRNA. What are code mRNA? We call it as codon lah. Codon, kan? So every 3 bases it code for specific amino acid. So there are 20 different types of amino acid. So according to the base sequence of the on the mRNA, the tRNA will bring specific amino acid. Okay, adding the tRNA, kan? Adding the nucleotide one by one according to the code on the mRNA. So this is the polypeptide synthesize that will fold into specific shape. Kita belajar dulu kan, the function of the Golgi apparatus kan, modify the protein and then package into vesicle, and then the vesicle will fuse with the membrane and then secrete out the protein out of the cell. Itu proses dia lah kan. So the polypeptide will be folded into specific shape by the ribosome and then packaged into vesicle, secreted out of the protein. So this one, it becomes the enzyme, for example. So tak semestinya secreted out. So the enzyme can be used within the cell, kan? So this is the enzyme produced, enzyme A, that will involve in the pathway that lead to the synthesis of red pigment that cause the cell to be red. Okay. So the process of producing the protein, this one we call it as translation. Translate the information which is copied by the mRNA. So we have here two processes. Transcription, copy the information from DNA into smaller molecule, which is mRNA, messenger RNA. And then the mRNA goes to the cytoplasm, to the ribosome, involved in translating the information into protein. Okay. So now we have looked at, there's two types of RNA, kan? mRNA and also the tRNA. There's another type of RNA, we call it as rRNA, ribosomal RNA. Ribosomal RNA is the component within the ribosome. Ribosome is made up of a combination of rRNA and also protein. Okay, that is ribosome. Ribosome, it is the component of ribosome, it is made up of rRNA and also proteins. So we know now there's 3 types of RNA: messenger RNA, tRNA, and also rRNA. Okay, so this is the overview of the relation between the DNA, the process of transcription, translation, leading to the synthesis of protein. Okay, so itulah dia, macam itulah. So nampak ni kan, so sekaligus saya dah terangkan Ini secara general, ini pun juga, ini pun genetic code tu tadi kan, codon on the RNA kan, and then yang ini proses extra kita tengok, and then the process of translation. So saya punya explanation tadi roughly dah explain the overview on this slide. Okay, so let's look at the detail, right? Okay, so types of RNA. RNA exists as a single strand polynucleotide and important in the production of proteins. Three types of RNA. The first one is mRNA, messenger RNA. Write in your notes, please. Messenger RNA. So it is a linear sequence of mRNA that copy the information contained in the DNA, which is gene specific segment on the DNA that code code for the synthesis of certain protein, okay, to be translated in the translation process to make protein. Okay, mRNA travels to the ribosome in the cytoplasm. You can add here, and in the cytoplasm for the synthesis of protein that occurs in the cytoplasm. So and then the next type of RNA is rRNA, ribosomal RNA. Okay, is a component of the ribosomes. And then the third one is tRNA, transfer RNA. Each different tRNA carries specific amino acids to the ribosome. We have 20 different types of amino acids. So therefore, we have different types of tRNA that carries different amino acid according to the codon, according to the information in the mRNA. Okay, so the definition of transcription. Okay. Saya share slide tak tadi? Saya share kan?
Share.
Saya share. Sebab dia tak keluar kat saya punya laptop. Okay, tak apa. Thank you. Okay, so let's look at the definition for transcription. So transcription is the synthesis of RNA using a DNA template. Okay, so the information from DNA is transcribed or copied into RNA molecule. Transcription produces messenger RNA. Okay, so transcription leads to the process which is translation. So translation is the synthesis of a polypeptide using the information in the mRNA. So ribosomes is the organelle that is used in the translation process. It is the site of translation. Ribosome is the organelle involved in protein synthesis. Okay. Okay, so let's look at the process of transcription and translation in a eukaryotic cell. So we are going to look at in eukaryotic cells first because eukaryotic is simple organisms. And so the process should be much more simpler compared to the transcription translation in eukaryotic cell. So let's look at the prokaryotic cell first. So this is, for example, a bacterial cell where the DNA is located in the nucleoid region, not in the nucleus. Okay, bacteria doesn't have any nucleus. The DNA is located in the nucleoid region. Okay, so therefore, the process of both transcription and translation both occurs in the cytoplasm. Okay, in the cytoplasm. So transcription occurs in the cytoplasm. Okay, so a gene provides the instruction for synthesizing specific mRNA lah. So let's say, for example, this is the DNA, kan, a segment of a DNA of a prokaryote. It has several genes, kan, gene 1, gene 2, gene 3, for example. Okay, so at this point, kan, at this moment, for example, information from gene 3 is required to produce certain enzyme. Okay, so only information from gene 3 in the DNA is required in the process of transcription and translation. Okay, so information from gene 3 is copied through transcription to produce mRNA. Okay, so this mRNA copies the information from gene 3 and then leads to the process of translation where the mRNA finds a ribosome, goes in between the large and small subunit of ribosome, and the ribosome reads the information of the mRNA to produce protein according to the codon on the mRNA kan. So the synthesis of the polypeptide chain is through the process of translation. Okay, so the translation, for the translation process, the information encoded in the mRNA determines the sequence of amino acid to form a specific polypeptide. So it occurs in the ribosome, at the ribosome in the cytoplasm. Okay, so for example kan, we have the mRNA. Yeah, so this is the, let's say lah, this is the mRNA here. Kan? So mRNA is single strand. mRNA are polynucleotide, so it has base sequence. So let's say lah, for example, the base sequence is AUG, GG, GGG, GCC, GAT. mRNA tak ada T mRNA, RNA doesn't have thymine, so thymine you replace with uracil. Solve pula itu. Kan? So these are the code kan, the code on the mRNA that will read in as codon kan. So every three bases we call it as codon. Kan? Ini G lah for example kan. Kan? So every three bases the specific tRNA will come and add the first amino acid, and then the second amino acid, and then the fourth, the fifth, the next one, and the next amino acid can according to the codon. So this produce a polypeptide chain according to the code on the mRNA. Okay, so let's look at the overview of this slide, then we finish off. So this is the difference between transcription, translation in eukaryotic cell. So in eukaryotic cell, because we have nucleus, kan, so the process of translation, it occurs in the cytoplasm. Okay, so transcription in the nucleus, and then the transcription gene provides the instruction for synthesizing the mRNA. Okay, so this is DNA. Only certain gene will be activated, will be involved in the process of transcription translation. Okay, so once the RNA produced, the RNA cannot be used straight away in the protein synthesis. This initial strand, we call it as pre-mRNA. Okay, so pre-mRNA has to undergo an extra stage, we call it as RNA processing, that makes the mRNA shorter. And this one we call it as mature mRNA. Then only this mature mRNA migrate or exported out of the nucleus, goes to the cytoplasm, finds a ribosome and involved in protein synthesis, which is translation. Okay, so translation, so the information encoded in the mRNA determines the sequence of amino acids to form specific polypeptide. So occurs at ribosome in the cytoplasm. So the difference between the eukaryote and prokaryote, it has this extra stage. Okay, and the process occurs for transcription, it occurs in the nucleus. So let's end our session for at this moment. Do you have any questions so far? Ada soalan? It is already 9:51. If you have question, you can ask. If not, we can end our session. Tak ada? Kalau tak ada, kita boleh end our session class. So thank you.
Thank you so much, madam.
Thank you, madam.
Thank you, madam.
Thank
you, madam.
Thank you,
Thank you, madam.
madam.
Thank you, madam.
Okay.
Thank you, madam.
Thank you, madam.
Bye.